on active matter, biophysics, and related topics. So today's speaker is Roop Malik. We will just introduce him briefly in a minute. But I just remind all the attendees that the talk will be in total around 1 hour, including questions. And you are all very welcome to ask questions to the speaker also during the talk. So please don't hesitate to participate and make this as lively as possible. So I leave the stage to Mitun.
Thank you, Edgar. So welcome, everyone. It's a pleasure to have Professor Ruth Mallik from the Biosciences and Bioengineering Department of IIT Bombay with us today. Roop is, of course, a famous biophysicist, so he's done pioneering work in the field of intracellular transport by molecular motors, in particular by Kinesin and Dyneem motors. He's been awarded multiple recognitions, in particular the Shanti Swarup Bhatnagar Award in Biological Sciences in 2014, and the Infosys Prize in Life Sciences also in 2018. Roop is a fellow of the Indian National Science Academy as well as the Indian Academy of Sciences. In 2021, since 2021, he's been an associate member of the European Molecular Biology Organization. So I think Roop is a particularly apt candidate for this series of talks. Roop started his career as a physicist in condensed matter physics in TIFR before moving to biology, first at NCBS and then at UC Irvine as a Human Frontier Sciences Program fellow. Since 2006, he had been at the Tata Institute of Fundamental Research in Bombay, and in 2020, he moved to IIT Bombay, where he is currently. So today, Roop is going to talk about molecular motors from physics to physiology. Roop, please take it away.
Thank you, Mitun, and hello to everybody. Whether you are in India or in Europe somewhere, good morning, good afternoon, whatever applies to you. Can you hear me properly and can you see my screen?
Yes.
Okay. So we'll get started. So this is how I named what I'm going to talk about today. And I will start with this little picture, which I took roaming around here in somewhere in Maharashtra, and I have shown earlier on earlier occasions. But I think this is a very apt way to start this talk because, because this is what you see here. A horse and a bull, which is a rather unusual, carrying something which you won't see here, which I will show you a little bit, a cargo, which is on these wheels along a track, is essentially what we are going to talk about today. Now, the reason this unusual picture, you don't often see a bull and a horse yoked together, but that's kind of what happens in the context of these motor proteins that I'm going to talk about. There are different kind of motors. So you can think of these as motors, basically, entities which transport other things. In this case, a man sitting here behind these motors, and there can be of different kinds. So therefore, the analogy to the bull and the horse. And if you go behind this, which happened when I passed this, these two objects moving on the road, you will see this man who is the cargo for this horse and the bull. And one of the important points, of course, we will talk a little bit about the horse and the bull, but an important point which has often been, less, investigated in this aspect is this kind of a spherical shell around this cargo. Okay, so this is the cargo, this guy sitting at the back, and this spherical shell is a membrane. Okay, we are going to get inside cells soon and talk about how things are transported inside cells. This, this membrane which surrounds this cargo, and the horse and the bull are somehow attached to this membrane. This membrane is a living object. It can change, it can have different physical properties, chemical properties, mechanical properties, and that will be of interest to us as we move forward. Okay. Now, I will see how much of this I can manage, but what I've tried to do is, talk about things that we have done in the past and move on a little bit to what we are doing in the present. And if time permits, we'll talk a little bit about where we want to go. Okay, so now this video, many of you might have seen, it shows what happens inside living cells. And now, just before I go on, I will start this video and pause it in a little bit. And yeah, you see this something called a vesicle here and this motor protein. So this object is, our center of attention today, but not just this object, also that membrane that I told you around the cargo. So the cargo for the motor is inside this object called an organelle or a vesicle inside cells, and it is bound by this lipid membrane shown here in blue, to which this horse or this motor protein is bound. It is important to keep some idea of the dimensions here. This object, this motor here, is of the order of 100 nanometers. So we are really talking about nanoscale machines here. The cargo can vary in size. It can be about a micron, or it can be even much larger than that. So that's a rough idea. Now, these motors, as you can see here, as they walk in steps like this guy is doing, and as they step along, yeah, so you can see the steps here in this animation. As they step along, inside each head, it can burn some molecules of adenosine triphosphate, which is how you package the food that you eat into energy. And the chemical energy released from that adenosine triphosphate is converted into mechanical energy, which is manifested in the form of these repetitive steps as this motor protein walks. Okay. Now, it is also important to just realize a few things here. Each of these steps is of the order of 10 nanometers. It can change again. And this is really fast because a step can be taken approximately within millisecond time scales. Okay, so this is a really fast enzyme. And obviously, if this motor or this horse or this bullock, whatever you want to call it, it has to carry this object. It has to do some work, and it has to generate some force. These forces are of the order of piconewtons. And for us in our daily life, life, the forces of newtons or tens of newtons are more common. So these are really million times smaller than the forces that you and I can exert. And I won't go into the details of these things, but one can measure the efficiency of these single motors. They're approximately 50% for kinesin, and this number can vary for other kinds of motors. Now, let me also, just for the sake of completion, say that there are many different kinds of motors inside cells. There are motors which sit at one place and turn round and round like an electrical engine a little bit, you can think of it. And those are rotational motors. What you see here, what we are going to talk about are linear motors, which are linear because they move in a linear path like this. And we are going to restrict ourselves to linear motors, which walk on these kind of filaments, linear filaments called microtubules. There are other kinds of motors which walk on other kinds of filaments, but we will keep the discussion simple today. Okay. Now, what are these microtubules? These are polymers. Okay, so you take a protein, and you add another protein, repeats of the same protein, and make a long polymer, which you can see here in this fluorescence image. This polymer here is labeled in green. Okay, and you can see these are microtubules, okay, small tubes, you can think of them. These are spread all across this cell, where this blue is the nucleus of the cell, and these microtubules start somewhere near the nucleus usually and spread out everywhere. And this is practically true for all kinds of cells that you think about, okay? And again, just to keep in mind, this motor itself is of the order of 100 nanometers, less than a micron, but these distances in this particular cell is of the order of 50 microns, but in other kinds of cells in your body, let's say the neurons, this distance can be of the order of meters, okay? So imagine something which is, roughly 1 billion times smaller has to carry a cargo across the distance of meters. That's, not an easy thing to do. Now, because I understand that some of our colleagues come from a theoretical background, I will just very briefly talk about this. This is not something that we directly work on, but to understand this stepping behavior, one of the ways in which you can think of it is a ball and ratchet sort of model. So this microtubule that you see underneath, on which this object guy is walking, okay, this is made up of repeats of a protein, as I told you. And the protein here is charged. It has a charge. It has an electrical charge. Okay. And these legs of the motor, as it is stepping along this, that also has a certain charge. So when the motor comes close to this filament, it sees a potential, series of potential wells. And one can, based on experimental evidence, which I will not go into right now, this is an asymmetric potential well, as you can see here, right? There is, one side, one side is almost vertical and the other side is gradual. So what you can think of this as a model where a ball and ratchet, where what adenosine triphosphate does is that it provides the energy to raise this leg away from this microtubule, and once that happens, this motor really cannot see this these potential wells very clearly, because it has moved away from this this charged object, and then the motor basically diffuses around, okay, for a while. And it because of the asymmetric nature of this potential well, once it diffuses beyond this point, there's a higher probability that it will go to this side and not to this side. And therefore, this is a biased thermal ratchet in some ways because of the nature of these interactions. Okay, it is called a flashing ratchet because it flashes on and off depending on the distance between this and this. Okay. If there are any questions at this time, I can take, otherwise I will move on. All right. Now, what you saw here was one single motor carrying this large cargo, but that is unfortunately, like in all kinds of biology, in biological problems, that is not what happens inside cells. And what is important to understand, if I take a cargo, okay, so this is a typical cargo, I mean, it's of course an animation, a cartoon, but this could be mitochondria inside cells. It could be, different kinds of things, virus particles with a membrane around it. It could be any foreign object that has been taken inside cells. The surface of that cargo will look a little bit like this, okay, and with many kinds of proteins attached to this membrane, which is shown here in green. And these proteins could be motor proteins, ATPases, various kinds of proteins. Now, for the purpose of this, because we are interested in the motors, so the reality is that on a given cargo, and I will show you an example here. So these are mitochondria, okay, which is the cargo for these motors. And this is how mitochondria move in uninfected neurons. So just to give you an idea, these straight lines that you see are the axons of a neuron. They are the projections of a neuron. And if you look carefully again in this, the mitochondria, which are these objects here, they move in a back and forth stochastic manner. And if you now look at the same, the neurons which are infected with the rabies virus, you will see something which, at least to me, is quite scary. Okay, so this is how things look when you are fine, and this might be what happens when you are bitten by a rabid dog. Okay, so things just stop moving, and so there are a lot of disease relevance to how these motors move and how virus can hijack these motors, and in many other contexts, but that's something that we'll not go into. But to explain this kind of back and forth motion, one of the obvious ways to think about it is that given a mitochondria or any kind of cargo inside cells, which is walking along this microtubule here, okay, you have different kinds of motors. Remember the horses and the bullocks that I had shown, but these horses and these, yes, Mithun, you have a question?
Yeah, I was just wondering, so when you showed this infected neurons, is it that the motors do not attach, or is it that the motors attached but they cannot move the cargo?
So there is no one answer to this, Mithun. There can be many different ways. So the virus can change its, its code itself so that the motors can't attach. The motors might attach that, but they might get inactivated in some way. So it's very often we don't know the reason, honestly, but there is no one answer to this question.
I see. Okay.
Yeah. So now, and maybe I will touch on that part a little bit because you have asked this question. And so the reality is that given, let's say, mitochondria, what you saw here, moving around, there can be different kinds of motors, the horses and the bullocks that you saw. And these motors, let's say the green ones and the orange ones, they might not all want to move in the same direction. That's why you see these mitochondria moving sometimes this way and sometimes that way. And this is characteristic of many kinds of cargos inside cells. Okay. So the one class of motors, the green ones, let's call them the kinesins, they generate force in a particular direction along the microtubule. So when I say that, it is important to understand that the interaction between this part of the motor and this filament, they can recognize the polarity in this filament, and they know that I need to go towards one side of the filament and the other guys want to go in the other side. Okay, so they typically, these two classes of motors, this will be important to remember, the dyneins and the kinesins generate antagonistic force. Okay, one wants to go the other way and the other wants to go the other way. Now, given such a scenario, many questions arise, obviously. What are the forces that each of these green and these orange guys generate? What are their numbers? I've just put, this is just some imagination where I put 3 of each, 3 and 4 of type, but this may not be true for all cases. How long can these motors, given a motor, how long can they generate force? Okay, can they just keep generating force forever? What is their tenacity? How compliant are they mechanically? Okay, because that decides the configuration of these motors on this cargo. Are there other proteins? As you saw in this picture, there are so many other proteins other than the motors. Do other proteins control these motors in some way, which we sometimes understand, sometimes we don't understand? And lastly comes to the point of geometry, okay, which may be more interesting to this audience, where, we are not sure that the motors are attached in this kind of random geometry that I show here. Maybe they're attached in some other ways, and maybe biology can make use of that. Okay, so these are some of the aspects that I will touch upon. I will not be able to go into great detail in anything because of shortage of time, but if you now start looking at the literature, if you try to start understanding this problem, what you will typically see that if I zoom in into one of these motors, let's say I pick this Dyneem motor and I zoom in into this and try to look into the literature, what I will see is that here is a cargo, let's say the cargo here, and along with this Dyneem here is right here at the end. Okay, this is Dyneem. This is the business end of things. Okay, this is the one where energy is generated and force is generated. But before you reach the cargo, there are these, 20, 30 different, all kinds of proteins which are attached to this. Now, all of these proteins, the additional proteins which seem to be attached to this motor, are obviously very important because mutations in these proteins cause various kinds of diseases, and we need to understand this. But the reason we know so much about these interacting proteins is because typically the people who work on this, people with a biology background, these techniques of finding which protein interacts with something and how it can be broken and trying to make mutations in these proteins, these techniques are very well established, and therefore over many years of really pioneering work, we have figured out something like this, what you see in this picture. Okay, but if you are coming in as a physicist and if you want to learn about this problem, when you see this picture, this is what this is what your response will be immediately. Okay, and then for many of us who are coming in from a more physics-oriented background, you look at it for a little while like this, and then you give a deep sigh and you move on to something else. Okay, But is there something that we can do? Okay. Now, in that, so when we talk about all of these proteins, which finally connect the motor here, the dyneem here, to this cargo, what are they doing? Okay, and one of the obvious things that they could be doing is that they are playing the role of a traffic policeman. Okay, which depending on signals, sometimes which we understand, sometimes that we do not understand, they can stop this motor or activate some other motor. And because this traffic policeman is the person who finally decides what how these mitochondria will move. Okay, so imagine the red light being switched on and the green light being switched on and the yellow light being switched on, and that's why these mitochondria move like this. Well, that is quite true for many situations, and there are reasons to believe that, which I won't go into. But departing from this viewpoint, many people thought, particularly people coming from a background of a more physics or mathematics background, thought that can we think of this in a little bit different perspective. And what this, I would say, very influential paper by Leposky's group showed a while back is we can think of this back and forth motion as, let's say the kinesin and the dyneem motors attached like you are shown here. And there are stochastic transitions where these individual blue or the yellow motors have specific on rates and off rates. And because of that, given an on rate and an off rate and a certain force that they can generate and a certain number of these motors, you can predict how the cargo will move, perhaps in a back and forth manner. So in other words, you don't need this entire complexity to try to understand everything what motors do. Now, remember that we are not talking about single motors. We are talking about teams of motors. There are multiple blue motors and there are multiple yellow motors here, and how they will, finally, what will happen to this cargo, how their activity will manifest. That's something which can perhaps be explained by just think of these as stochastic transitions. But remember that this was a purely modeling sort of paper where they picked out on rates and off rates from other experimental work, and they just put it in the computer, and they modeled this work. This 2008 PNAS paper got quite a lot of attention. The same group published another paper around the same time, which which perhaps not so many people read. But what was really interesting to us in this other paper, sorry, I didn't put the journal name here, I should have. But if you can plot a phase diagram, okay, what do I mean? On this x-axis, I plot the number of kinesins, let's say the yellow guys, and on the y-axis, I plot the number of dyneins, the blue guys. And if I now model their motion, I can divide it into different categories, where this plus means that the motion is dominated by kinesins, minus means that the motion is dominated by dyneins. This red one means that nothing happens, the cargo doesn't move. That is like what you saw after after rabies infection. Okay, something like that you would see in this regime, where nothing actually moves, and in the various regimes. But what was very interesting to us was this narrow regime, okay, this green part that you see, where there would be transitions between one direction of motion and the other direction of motion, and interrupted by brief pauses, okay. And this happened when you had about 4 to 5, 3 to 4 kinesins, about 1 to 2 kinesins opposed by approximately 5, 6, 8 dyneins. Okay. Now, this model where the stochastic transition, there was a lot of improvement of this model. There were a lot of criticism, equal load sharing. The loads would not be shared equally by motors, and the force velocity curve, how the motor responds to a backward force when it is being pulled. These things were refined later, and very, very nice work was done by in 2 papers, and others. It's reviewed here by Amrish from our department. If you're interested, you can take a look at that. Okay, but I will not. Now, on these lines, we what we did. Remember, but this was this was a modeling theoretical work. We wanted to test some of these parts and see what happens in real experiments. Okay, so what we did is that you remember this object that was moving around inside cells, right? We pulled such objects along with the motors out of cells. Okay, and the polymer on which these motors walk, we stuck the polymers on a cover slip. And now if I run this movie, I think I don't have to tell you that this looks like a tug of war. Okay, it's obvious from this that that there are, look, this is part of the cargo and this is another part of the cargo, and one motor is pulling it along this microtubule in one direction, this membrane is stretching. Then this one guy gives up. Okay, then this guy holds on, and then you go in the other direction. So these are actually endosomes, which are when small objects or liquid material is taken inside cells, they get into these kind of compartments. And endosome biology is a completely different field, a very interesting field of view, which I don't want to get into right now. But what we could show on these kinds of cargos, endosomes, from our studies, was that the very interesting point that came out, that there is a competition between these motors, the kinesin motors and the dyneem motors here, but this competition is asymmetric. Okay, you don't have the same number of kinesins and dyneins. You rather have a large number of dyneins teamed up against 1 or 2 kinesins, which is exactly what you saw here in this space diagram. 1 to 2 kinesins teamed up against a large number of dyneins, and this creates a situation of force balance. This guy is strong. Okay, like a Superman we have shown here. This Pn means piconewtons. So it generates about 5.5, 6 piconewtons of force. But these guys are individually weaker, but together they can generate a large force and match this. And therefore, there's a force balance, which is what you need in a tug of war, obviously. Okay. Now, this is not, we don't believe that it's a trivial observation, because this can explain how do you control things inside cells? If something has to be taken towards the periphery of the cell or towards the center of the cell, you need to change things. What you can do in this model is that you can add and remove these weaker motors, and therefore you can bias the transport towards this side or towards that side. Okay, so that we think is biologically important, and I will come to a little bit more of that. What we went on to do further is we showed that this entire complexity, okay, that who controls all this, and how does this, who decides who goes when, the dynein or the kinesin. Remember that it is important to understand this, because this opposing activity of dynein and kinesin to a large extent decides what is going to be where inside cells. Okay, it's not just a mishmash of soupy things inside cells. Some things are present near the nucleus, some things are present at the periphery of the cell, some things are present in the middle. And this distribution, this polarized distribution of objects inside cells is absolutely important for cellular function. Okay, things interact with other things at certain places inside cells, and if that doesn't happen, the following biological processes cannot take place. So it is important to understand how, perhaps in this kind of a situation, whether you will go this way or you will go that way. Okay. All right. So what we were able to show, along with along with help from Ranjit in our department, is that this entire decision between kinesin and dynein can be explained, and these were based on experimental data, and we analyzed the data with a simple binomial distribution model, where we could show that the choice between dynein or kinesin can be attributed to the simple tossing of a coin. Okay, the coin, you get a head, you decide on dynein, you get a tail, you decide kinesin. And so what we could find in agreement with the Lipovsky paper, again, this was by interpretation of experimental data, I'm not going to go into the details, is that this coin, for the particular case that we were studying, was a fair coin. Okay, so 50% of the times you choose dynein, 50% of the times you choose kinesin, and that made sense because the object, the cargo that was moving, was supposed to be not going anywhere, in the long run. These were early endosomes. They are moving in a back and forth manner, so you need a fair coin. Okay, sometimes you go one direction, sometimes you go the other direction. And the hypothesis and the implication, therefore, is that the proteins that associate with these motors, remember all those complicated things which link the motor to the cargo, they can make this coin unfair, okay, when it is needed. And what we believe is happening is that you decide a certain number of motors on a cargo, let's say, something like this, 5 versus 1, and the enzymatic properties, the on rates and the off rates of these motors, their strength, their tenacity, they decide the fairness of this coin, whether you will go this way or you will go this way. Okay, so on the short term, the decision between the dynein and the kinesin is decided by the tossing of a coin. Now that coin may not be fair coin. Okay, it can be an unfair coin, and that fairness is perhaps controlled on the long term. When the cell wants to change the polarization, it can make the coin unfair, and that is where all these proteins come in, which control the fairness of a coin. So hypothetically, you can think of this problem as the tossing of a coin in a simple way, and that the fairness of that coin is the only parameter that you change because of all the other paraphernalia that controls these motors. Now, to understand this from this point of view, there are many important parameters, and this is an incomplete list, of course. The surface density of these motors on the membrane is important. Their on rates and the off rates are important. The geometry by which these motors attach is important. And we have picked up some of these aspects in later work, and I will just show one slide each before I move on to some other things. Now, when I talked about all of these things, on rates, off rates, in the numbers, I told you there are 5 or 6 versus 1. How do we actually measure all this? Okay, so we can measure these things fairly precisely, and this is not because of methods that we have developed. These methods have existed for a long time, and one of the ways of doing it, of course, you have to combine this with many other biochemical experiments, which I'm not going to go into, is an optical tweezer. Okay, so an optical tweezer is a focused laser beam. As you switched it on, it's a focused laser beam like this, and you typically build it in a microscope. And you can trap small. So here is this focus beam. So imagine where my laser pointer is. That is the focus of this beam. And you can trap small refractile objects that bend light at the focus point of this laser beam. I will not go into the details of how that trapping happens, but the important point is that so you can take a cargo, in this case, a simple plastic bead, okay, and attach a motor to this. And when the motor pulls the bead out of this focused laser point, so the motor applies force in this direction. Here is the motor, okay, and here is the motor. When the motor pulls this out, this trap works like a spring, as is shown here, and it pulls the object back to the center. So that's what you see happening. Okay, so there's the motor pulling it out. You start again. You pull it out. Come on. Yeah, so this object gets pulled out. You can see these blips here, okay? And now it will again get pulled out, and then it will fall back to the center, and this kind of blip. So this distance along this axis is how far you move. That is plotted here. X, okay, along this X direction as a function of time here, and this is the position of the cargo in the optical trap. And as you move away from the center of the trap, you can imagine that you're stretching a spring more and more, okay, and somewhere a balance will be reached where the force from the motor is equal to the restoring force of the spring, and there you can't move any farther, and eventually you give up and you fall back to the center. Okay. Now, this kind of data, and when this kind of events will repeat again and again and again. Okay, this is a rare opportunity to see an enzyme live in action. Okay, you can measure on rates, off rates, tenacities. You can get a wealth of data from these kinds of records. Okay, but what we typically, these kinds of measurements have been done for decades now with plastic beads that have been coated with motors. So you are not really understanding what happens in biology. You are understanding an object of your own creation. Largely, you are trying to understand how single motors work, and we have gained a lot from that. We did not want to continue doing that. Okay, when I started my lab, I wanted to understand something closer to biology. So what I thought, based on work done by earlier people, is that what we can do, we can just grow cells on a cover slip and we can feed them with plastic beads, the same beads which are shown here. Okay, so these cells, they can be cells of your immune system. They recognize these beads as foreign particles, let's say bacteria. Okay, and they will eat these beads. And now, if I look inside a cell, this is what you will see. Okay, these beads are inside the cell and they move vigorously. Okay. And so what is going on is that once these beads get inside cells, they acquire a membrane around themselves. And all these dynein and the kinesin motors assemble on these membranes in a manner that comes naturally to the cell. So these beads can now, once you have let them inside the cell for a while, you can burst these cells open and extract these beads. And these are called phagosomes. Okay, you have to remember this. These are called phagosomes. These beads, they're called phagosomes. Okay, so rather than studying an artificial object, which is a plastic bead to which you have somehow managed to bind a motor, you have something like this. Okay, which is a phagosome, which you have extracted from the cell, and you can study this. You can take it out of the cell and you can put it on a microtubule, like what I was showing you earlier, and you can try to understand what happens in a more natural situation. Okay, so now going back to this problem of these two kinds of antagonistic motors, the green ones and the kinesins and the dyneins, what earlier people had done by using plastic beads coated with kinesin, that they had shown that the kinesin motors work fine when they're working in singles, like we are seeing in this cartoon. But if you put two kinesin motors together, they don't do much better than a single motor. Okay. In other words, two kinesins transport cargo primarily via the action of one motor. Okay. So it is not very useful to build large teams of this kinesin motor, because if you take 2 or 3 or 4, the function does not improve much. In contrast to that, what we were able to show that is absolutely not true for the other motor, which is called dyneem. Okay, remember this cargo where these orange guys are the dyneins and the green ones the kinesin. And I had also shown you this cartoon earlier where we had some 6 or 7 dyneins teamed against a few kinesins, and that's what is shown here. The dynein motors, when you put them together, if you have 1 versus 2 versus 3, so this is the force that they generate, and this is the time for which this force is generated. You can see that if you go from 1 motor to something like 10 dynein, 1 dynein to 10 dynein, the amount of force that they can generate, which is on the y-axis, increases tenfold, and the time for which this force can be generated also goes up. Okay. In other words, the dynein motor is able to cooperate. These motors are able to cooperate. And this is really interesting because there are many kinds of cellular processes. For example, when the when the brain of a child is developing and the neurons have to move to the right place inside the brain, it is the dynein motor which is predominantly responsible for moving such heavy cargoes. Heavy in the sense that, against opposing load. So this was very interesting to us, that if you simply tune the number of dynein motors inside cells, you can get a completely different function, which is not true for at least some of the kinesins that we have seen.
Excuse me, Roop, can I
Yes.
ask a clarification? So on phagosomes, do we have a control of how many motors or what type of motors to attach, or is
We
it?
don't have a control. We don't want to control it. Okay. We want the cell to attach the number of motors that it normally does. Whatever it does, we can measure those numbers.
I see. So after you get this, you can identify which motors or how many of them are attached.
Yes,
is that right?
we can,
Okay.
because we know which direction it is moving in. So we therefore assume that it is dynein or kinesin.
Okay, thank you.
But in principle, if we wanted, we could control the number of motors, but we deliberately didn't want to do that because we wanted to understand what happens in a natural way.
Okay, thanks.
But you're doing the experiment outside, right? I mean, they were initially inside the cell, then you burst it, and now they have come out. So you can't control the ATP or I don't know, whatever else.
So I did not go into the details, Apratim, but we have done these measurements inside cells also.
I see. That's interesting.
Okay, so some of the measurements were done outside cells. What you see here, so let me explain. We can actually send in an optical trap where you see my laser pointer inside the cell and trap one of these guys. Okay. And there also we can do similar kinds of measurements, which were yeah. So in this paper, you will see that.
I see. Okay.
And we don't see anything hugely different between what we measure inside cells and what we measure outside cells.
Okay. And
are there any other questions?
Yes. So suppose there's a cargo and
can you speak up a little louder, please?
Yeah. So can dyneins and kinesins cross each other while walking on the same microtubule? I mean,
yes, they very likely can. Nobody has actually seen that happening, but because these microtubules have multiple protofilaments, they should be able to cross each other in principle.
Oh, so dyneins and kinesins work on different protofilaments. That's why you expect them to be able to cross protofilaments.
There's nothing stopping them from working on different protofilaments. Whether they actually do or not, I think they do.
Okay. Thank you.
Yeah.
At the moment, yes.
Yeah, it's like a multi-lane highway. Okay. So now, yeah. So this is what a cartoon of the dynein motor looks like. It is trying to move in this direction. So this is the minus end of the microtubule, and while it is carrying something, let us say, okay. So this, there's a lot of thermal oscillation, and what happens is that inside each of these, legs of dynein, you hydrolyze ATP here, okay, and you that energy is converted to mechanical energy, and you keep stepping like this, okay. So what we were able to show a while back, based on our results, is that inside each head, inside this object here, there is something like a gear-like mechanism, which is what I've tried to show you with these wheels turning. And this gear is actually controlled by the binding of adenosine triphosphate inside this nanoscale head. Okay, so it's essentially a nanoscale gear-like mechanism which nature seems to have designed. And why is that useful? Of course, so we found that in 2005, when I was doing my postdoc, we figured out that something like this might be happening inside each head of dynein. This is a geared car, which is unlike the ungeared scooters that you see. But why is such a gear useful? Okay, some hint of this we got because you remember in the few slides ago I told you that this dynein works very well as a team, is what we were able to show. We believe that this gear allows dyneins to work in a team. How might that happen? Now, there is some evidence for this. I'm not going to the details of this, but I will just show you one slide and move on. Is that imagine that you are a cargo here and there are 4 dyneins shown here in green, and you are kept in an optical trap. Okay, so when you try to move out of the optical trap, you will experience a backward force from the trap. Okay. Obviously, normally when you are inside cells, you don't have an optical trap, but that backward force in the absence of an optical trap will come from kinesin, which is trying to pull you in the opposite direction. So when 4 dyneins try to move towards the right, as they move farther and farther, they're pulled back. what happens is that the dynein at the front shifts into lower gears, okay? As I told you that there's a gear-like mechanism which senses load and it goes slower, okay? So you know what happens in a gear, that if you want to climb uphill, you go into lower gear. So dynein, just like that, when it tries to move against load, wants to go uphill, it shifts into lower gear and it reduces the size of its steps, so it slows down its velocity. When that happens, the dyneins at the rear don't see such a high load, so they continue taking large steps. So whenever they are separated from each other stochastically, the normal tendency is for them, for the ones at the front, to slow down and wait for their partners to come and catch up with them. So dyneins normally, therefore, what we thought, work well in a team because they don't get separated too far because of this gear-like behavior. So the analogy is that if you take multiple cars and bind them with a semi-flexible linkage and you ask them to climb up a hill, geared cars will do better than ungeared cars. Okay, it's a rough analogy, but I think I will move on to something else. Now, this teamwork in motors, how well do motors work together, is not just something which you investigate and it's for the sake of fun. It is actually can be a matter of life and death. Okay, why? Because you remember this picture, this movie that I showed you. This is an immune cell of your body. These are the kind of cells that you have in your lungs, in your kidneys, in your liver, all places. These are the cells which protect you from all kinds of nasty things that could happen to you. And what could be such a nasty thing? A bacteria, let's say this laser pointer, coming into this cell, and the bacteria would get trapped into such a phagosome. Instead of the bead, you would have a bacteria inside, and that's what I've tried to show you here. Okay, so here could be a bacteria trapped inside a phagosome, and on the membrane of this phagosome, you have these dynein motors and also the kinesin motors, which I'm not showing here. Okay, so this bacteria is happy. Now, what happens is that as time passes, this phagosome moves back and forth, back and forth. Eventually, I'm not going to details of that. What happens is that the amount of cholesterol on the membrane of this bacteria increases. Okay, cholesterol is a hydrophobic molecule, and along with other sphingolipids, it assembles into these kind of patches, which are shown here in red. And the dynein motors prefer to sit on cholesterol. So you have all the dyneins that were distributed uniformly here, they get recruited to these small patches. And once that happens, and if one of these patches is oriented towards the microtubule, you can have lots of dyneins sitting on this patch, which can generate force along the microtubule. And you can take this bacteria sitting inside this towards the center of the cell, towards something called a lysosome, where you're supposed to kill the bacteria. Okay, and you can actually see this clustering here in this picture, where at the early stage, when you just enter the cell, the dynein shown in red is distributed uniformly on a phagosome, but at the later phase here, you see it is in these kind of patches here. Okay, and this is where you see more cholesterol also. Okay, so cholesterol is not all that bad. Okay, and this work was done very capably by Ashim, Divyan, others in the lab. Now, why is this interesting from a physics point of view? If there are Italian friends, I particularly thought this slide would be interesting to them. So if you take a cargo in the form of a cricket ball, and our cricket ball for our Italian friends is something which would fit nicely into your hands. It's a small cargo. And if you have motors of this size, okay, roughly think of them as cigarettes, okay, if you attach motors to this, then however many motors you attach, only half of them will be able to reach the microtubule, right? Because the dimensions of the motor are comparable to the size of the cricket ball. And you can actually, there is a particular patch. So you can see here, there is a particular patch here, the area at the bottom of this from here where you can reach this microtubule. If your size is, let us say, this much, and if you are sitting here, then you can't reach the microtubule, right? So given a size of the motor, there is a particular area at the bottom of the cargo close to the microtubule shown here in red. And if you look from the bottom shown here in gray, from where you can access the microtubule. So if I randomly throw motor proteins on this cargo, what is the probability that I will hit this area of contact? Let us calculate that. That's easy enough to calculate. You know the dimensions, you can do a little bit of geometry. And you consider the probability of contacting a microtubule is the area of this divided by the total surface area. And that probability, so this is the probability if I throw a motor on a cargo of radius r, what is the probability that it will be able to contact the microtubule? It goes down rapidly. Okay, it's a square dependence. You can see that here as the surface of the cargo increases, surface area, the cargo size increases. In other words, if I have large cargos, if I want to use the same motors to move a football rather than the cricket ball, adding motors randomly to the surface of this football is beyond a certain point useless. Okay, rather I should add motors to certain patches on this football, let's say the black parts, so that you can have a higher chance of this patch contacting the microtubule. And such patches, I won't go into the details again, are known as lipid draft, cholesterol-rich lipid draft domains, which have been demonstrated in many other contexts, mostly in artificial systems. But we could show one use of why such a lipid draft, cholesterol-rich lipid draft, may actually be useful inside cells. Okay. Very quickly, what we have since been in a more recent work, Paulomi and Pankaj very nicely showed that, so you remember these orange motors, the dyneins. Now you will see this little prickly thing which is shown here. This is one of those proteins which attaches with dynein. Okay, so here in this animation, let's say this is a cargo, and here is dynein which is walking along the microtubule. To it, attached to it, it has this dynactin. So remember, dynein activating protein, dynactin. And if I now think of grandpa walking with a stick, it is a little bit like that. Okay, so here is dynactin. This is the stick. Okay, and here is dynein. He's generating force, but he needs something for stability to stay on the microtubule. What we were able to show, we could specifically disrupt this attachment between dynein and dynactin, or this attachment between the stick and the microtubule. We could disrupt these things specifically and show that a particular part, this is a complex, of proteins, okay, which actually does something. And we could actually show by measuring the enzymatic activity of these proteins, if you disrupt this interaction between the hand and the stick, which is this one here, you specifically prevent the motor from getting on. But if you disrupt this one, you specifically prevent the motor from coming off. Okay, so in other words, we could show on a complex of proteins in a natural environment that there are separable on and off controls. Okay, and now this is why is this important? Because there are many mutations in dynactin. Okay, when you change this protein in response to many diseases, where this specific, the off rate of dynein increases, means that the dynein-dynactin complex cannot generate force persistently. This is true for several kinds of mutations which lead to neurological disorders. So it is really important, we think, to be able to separate these two controls and to understand how these things happen. Okay. I have a little bit of time, so I will touch on something completely different that we work in the lab, which is related to digesting fat. Okay. Now, here is, let's say, a cargo, let's say a phagosome inside a cell with this membrane around it. And you will see that this membrane is a bilayer membrane with a hydrophobic part consisting of fatty acid chains in the middle and phospholipid groups at the edges. Okay. And you assemble into these kind of stable structures, which can be the covering membrane of a phagosome. And to this, you attach all these kinds of proteins, motors. Some motors are shown, non-motors. Now, there's another kind of cargo inside the cell, which is fundamentally different, which is called a lipid droplet. And basically, all this stuff that you eat goes and gets stored here in a fatty hydrophobic environment inside this lipid droplet. And the obvious implication is that unlike this bilayer membrane of your normal garden variety vesicles, these lipid droplets because the inside is hydrophobic, they have a monolayer membrane where this fatty acid chain can happily sit in this hydrophobic region. So this is what a lipid droplet looks like. And you can stain these lipid droplets with various dyes, and you can see that they move around very often in a crazy manner, and we don't understand why they're moving. Okay. So that is one thing where what is the purpose of this motion of lipid droplet? So why is motion of lipid droplets important? Because they are the site where you store energy and membrane inside cells. Okay. And in response to different metabolic states, different kind of infection, different kinds of diseases, obesity, diabetes, the number of lipid droplets inside cells can change. And very often these diseases arise because the fat stored in these lipid droplets is not being exchanged properly with other components of the cell. Okay. Now, come to think of it, one of the things that, is a very huge problem in our society and in Western society also is the accumulation of fat inside the liver. Okay. It's called fatty liver disease. In this context, what we were able to show a mechanism, a completely, we believe is a novel mechanism by which fat is cleared from the cells of your liver. Okay. We were able to show, I won't go into details of all this, but basically when you eat and the insulin levels are high, insulin activates a cascade of events which ultimately leads to a kinesin motor being recruited to these lipid droplets inside the cells of your liver. And when kinesin is attached to this, these lipid droplets will rapidly be transported to the periphery of the cells. This is a single hepatocyte, single liver cell, to the periphery of these liver cells, where the fat inside these lipid droplets can be digested by enzymes present on the smooth endoplasmic reticulum, repackaged into something called very low density lipoprotein and secreted out into your blood. And when you go for a blood test, this is what you measure as serum triglycerides in your blood report. Okay. So the amount of serum triglyceride, if it is very high in your blood report, then that is bad news for you. But the fundamental mechanism behind this is that, this lipid droplet needs to come to this smooth ER and needs to get processed like this. So if we look at the end point now, we are moving to something different away from motors. Here is a lipid droplet, let us say, which has just reached this smooth endoplasmic reticulum, okay, here, and you will have the formation of such a geometric structure, which is a neck-like region, and the physics happening at this interaction region, the lipid droplet and this bone. So remember, this is a monolayer membrane, this is a bilayer membrane, okay, you can see that is really interesting. And not just from the physics point of view, of course the physics is interesting, but the consequence of this lipid droplet attaching to this leads to so many downstream processes that it is incredibly important to understand. Okay, but how do you understand this? It's really difficult because these structures are really, microscopic structures inside the cell. Their geometry is complicated. So what we thought is that this endoplasmic reticulum, smooth ER that you show here, or the endoplasmic reticulum in general, can we simplify the geometry? Okay, this is a very complicated structure, the ER membrane. Can we deposit the endoplasmic reticulum membrane in a planar geometry on a cover slip and think of some way of mimicking it? We were actually able to do it. Sukrut and Jagjit have done a fantastic amount of work where the endoplasmic reticulum from the liver of a rat was purified and it was deposited. I won't go into the details of this again. It was deposited on a cover slip. Okay, so you have a cover slip on which you have this ER, the endoplasmic reticulum is being mimicked here by this planar membrane. And now on this, so remember, here is the endoplasmic reticulum as a planar membrane on a cover slip now. And we take these lipid droplets, which are supposed to interact with this, and we flow them into the same flow cell in a completely in vitro system. And we let them be for some time. So remember that you are trying to replicate this process, okay, this lipid droplet coming and shaking hands with endoplasmic reticulum on a cover slip. And depending on the metabolic state of the animal, you can see that the interactions between this lipid droplet and the endoplasmic reticulum increase like crazy here, okay. And depending on the metabolic state, whether you are fed or fasted, that is where this insulin angle comes in. But I'm not going to go into the details of that. The other thing that we also saw is that if you have a bacterial infection, if you can reproduce that, then also the interaction between these lipid droplets and this planar membrane increases. And to do that, we use optical tweezers here to measure the strength of this tether. We could actually estimate the strength of this tether, this lipid droplet being tethered to this. We want to continue this work, and this is currently under revision. I think I will stop here. This, for the physicists in the audience, the force for this dissociation was in excess of 20 piconewtons, and which kind of measures the binding energy, which is basically the square of the line tension divided by the surface tension. So remember, this is what we are trying to replicate. And depending on this binding energy, if it is negative, you will be deeply embedded and you will be able to exchange material with the ER. If the binding energy is low, you will not be embedded. You will kind of sit out and will not exchange material, or you will not interact at all. We figured out that this entire process seems to have a lot of dependence on a conical phosphatidic acid, which is generated under the control of insulin, and that's why you can control how much this lipid droplet will interact with this. I will skip these parts because I don't want to be late. I will come to my last slide, where the first thing I want to do is, this is some of these members had come visiting, Mukesh and his wife, but this is the present composition of my lab. We are going in many different interesting directions. I did not have the time to talk about it. That's for the future, but I'm really proud and happy to be associated with a group of people, particularly because it has not been easy to start your lab again in a different place, but it's all because of their commitment and their interest that we keep going. And this is a picture of the beautiful IIT Bombay campus. I would like to acknowledge the people who fund our work. And I would also, even if you're coming from a theory background or an experimental background, there are possible openings for postdoctoral positions. Let me know if you're interested. I will stop there and I can take some questions if there are any.
Thanks a lot, Ruth. This is really
there's a lot of background noise.
Thanks, Roop. This is a really interesting talk. Anirban, if you want to ask a question, yeah, sure.
Should I write or should I just?
No, you can speak. You can speak.
Yeah,
oh, okay. So the last two slides basically. So that when that droplet is attaching to the
one minute. You're talking about this one?
No, sorry, one before this. Oh, sorry, one more. That
this
big, yeah. So this endoplasmic reticulum, this ACR, sort of taking in the lipid into it, right? So it looks like a endocytosis kind of thing.
No. It's not like that. Basically, what happens is that, so see, the lipid inside this is hydrophobic. It is triglycerides. Okay. They cannot just cross this membrane into this. They are converted into diacylglycerol, where they can go into this and then are converted back into triacylglycerol. Okay, it's not endocytosis. It's rather a membrane fusion, a transient or a less transient fusion. It's not endocytosis.
Okay. Then I had one more question. I mean, near the end, you showed that you found some difference happening when the lipids are conical type, right?
Yes. So this one. I will go to that slide. Give me a minute.
Yeah. So typically these kind of conical lipids, they render some kind of intrinsic curvature in physics term to the membranes.
Yes. That's what you see here. Hello?
Okay. Yeah. Okay. So you were showing that, but so as opposed to, I mean, if it was not conical, how would it would look like? I mean,
no, it would not be embedded so deeply in this one, right? If they were not conical, if they were all straight, then you would not be able to form this kind of curved regions.
Okay. I understand. Thank you.
Ashutosh, you can please unmute yourself and ask.
Ashutosh, you are muted.
Oh,
इससे म्यूट हो गया.
I just you talked something about insulin and the role of insulin in pinching of lipid droplet. I
Yes.
could follow quite that. So what is the role of insulin here?
Okay, so the role of insulin is, we believe it has multiple roles. One of the roles is that when insulin levels are high, you this protein ARF1, okay, it is activated. ARF1, once it is activated, it is in the GTP bound state. It has this prenyl group. It has this, I forget which group it has. Then ARF1 goes and binds to the lipid droplet membrane. Okay, that is one. Insulin is also known to activate phospholipase D1. Okay, once it is active, phospholipase D1 under the control of insulin, ARF1 is being activated and phospholipase D is being activated. This comes and binds to the lipid droplet. Phospholipase D converts phosphatidylcholine on this membrane into phosphatidic acid. So all of these things are happening downstream of insulin.
Okay.
So if you increase the insulin levels, basically you will discharge more very low density lipoproteins.
Okay, thank you.
So can I ask one more question?
Yes,
sorry.
I think there are some questions in the chat also. No, I think no, we have answered all this. Yeah, please. Yeah.
So you maybe you already answered this. So you talked about this this grown beads, phagosomes inside the
Yes.
cell.
Yes.
And so with this experiment, could you observe something that people couldn't do with the artificial beads, where
Yes, absolutely. The entire, so okay, that's a good point. Let me come to that. Why was using phagosomes useful, right? That's your question.
Yeah, exactly.
Yeah, I will come to that point. Yeah. So can you see this slide now?
Yes.
Yes. So these beads don't have a lipid membrane around them. Okay. The phagosomes do have a lipid membrane. So all this business that I told you about cholesterol and this, the formation of this lipid raft, it would have been impossible to do this with beads.
Okay. Okay. Right.
So we really learned something new about how biology does things, which was not so intuitive at all.
Okay. Okay. Got it.
Yeah. Nobody seems. If there is nobody else, I have one more question.
Yes, Anil.
Yeah. So you said that from the therapeutic side, if it becomes difficult for the ERs to absorb those lipid droplets, then I think you said that it will not go into the blood as LDL. Is that right? If I understand.
So let me try to explain. Let me go to that slide and let me try to explain.
Meaning my question is that if you prevent that, what would happen? Meaning it will not go to blood, but where will it go?
See, it is a delicate balance. It is a balance between how much fat you have inside your liver and how much fat your liver can manage versus how much fat you have in your blood. Okay.
Yeah.
So the question is, can we control this process in some way? There can be different kinds of people, patients, people who have fatty liver, let us say. Okay. What do you want to do for them? You want to reduce the amount of fat in their liver. Okay. So can you increase this entire pathway so that more fat gets sent out into blood? Right? Hello?
Yeah.
So that's one way of thinking about it. The other possibility is that the other kind of patient might be somebody who has too much serum triglycerides. Okay. And if you have too much fat floating around in your blood, it will go and get deposited in your heart eventually, in all kinds of places that will lead to cardiac arrest. Now, what we could show in this paper is that if I short circuit this mechanism that I'm showing you here, okay, then a huge amount of fat is secreted out into your blood every time you fast, which is going to happen every night when you go to sleep. Okay, and all that fat which goes, in the morning, all that fat which is now, if you short circuit this mechanism, it goes and accumulates in your heart. You were able to show that. Okay. So if you want, I can chat with you later, but it's hard to explain all this in such a short time.
Okay. Yeah.
Hello.
Thanks for now.
Yes.
I think Anil. Okay, sorry.
Hello.
Yeah, I can hear you.
Yeah. Okay. So, sir, I just need to ask that you showed one slide where you have shown that there is a need of war, like there is a tug of war between these motors. Like if these motors are sticking to one vesicle, so.
Right.
Yeah, so I just want to know that at a very normal time, why there is a need of this war. I mean, this is a waste of energy, right? If it is not moving in a particular direction, then what is the physiological significance then that they are
The
wasting so much
physiological
energy?
significance is that you don't just engage in tug of war, you engage in intermittent tug of war. It's not that you are pulling against each other all the time. Okay. Given the on rates and the off rates of these motors, you engage in tug of wars occasionally. And after that is done, you go in one direction or the other direction. Now, biology does not do anything because it wants to optimize energy. Biology has much more important things to do probably than energy. Okay. So if everything were energy optimized, then me and you would not exist. Right? I mean, we are breaking the rules of entropy whenever we are, becoming an organized system. So I don't think just looking at from the energy perspective is a good way to think about it. There are many other complex processes which have to be achieved, and for that you have to, use energy in the way that you have to. Okay, but the point is that this tug of war is not a continuous tug of war where things are pulling against each other all the time and not getting anywhere. These are intermittent tug of war. Tug of war, move. Tug of war, move other way, like that.
Sir, one more question is there. In those optical tubes, tweezer experiment, like how you people get to know that this is the minus end and this is the plus end, and on the basis of that you are putting either Dyneem or
we have ways of labeling the minus end of the microtubule using a biotinylated tubulin. I can discuss that with you separately, but that's a technical detail which I don't want to get into right now. We can do that. When you start growing these microtubules, you make a seed which has biotin attached to it. So that's
Anil, you can unmute and ask.
Yeah, hi. Can you hear?
Yes.
Yeah. Thanks for this great talk. I learned a lot. I have two short questions. First one is, I mean, there are a lot of these different types of motors, right? Like muscle motors, non-muscle motors.
Right.
I mean, the differences come in the terms of like the forces or the kind of a mechanism itself, like these different kind of motors.
Okay. No. Sorry, I did not get your question. What was the question?
So all these, are there, what kind of differences are there between these kind of classes of motors, like muscle motors and also these?
I see. So the muscle motors, essentially, they do not walk along microtubules, but they walk along another kind of filament called the actin filaments, okay, which are present in abundance in your muscles, okay. And the muscle motors work in even larger teams. I told you about teams of here 8 to 10. But imagine the amount of force needed for you to, carry some heavy object. There are millions and billions of these motors acting in synergy. Okay, but that is a completely different kind of structure where you have interdigitating filaments like this, and the motors work along them. So their geometry is completely different.
I see. Another short question is: are there any efforts towards making a synthetic motors?
Yes, people have done that. Yes, and indeed, a few years ago, the Nobel Prize in Chemistry went to such an effort. Yeah.
Oh, sorry. Thanks.
So, Antara, you can ask if you have a question.
Hey, Roop, it was a great talk.
Thank you.
So I just wanted to know, what do you think might be happening in case of, sometimes we see some organelles which get disorganized in some disease conditions. Like suppose in our lab, we study Golgi, which we see is disorganized in some kind of cancers. Right. So some studies show that dyneins are involved in keeping the Golgi organized. And alternatively, in the disease conditions, there is disorganization, which we do not yet know whether there might be a kinesin involved, or it could be because of the fall off of the dyneins. Right. So, but then in your talk, you have said that kinesins are not a very good team player. So moving such a heavy object
No,
towards...
but that doesn't mean there are many mutations in kinesins which lead to all kinds of diseases also. Okay. So whether you are a good team player or not, whatever effort you generate is required in some way inside cells. And having a mutation in kinesin can be equally bad or worse than having a mutation in dyneem. The only point is that there are 30, 40, 50 different kinds of kinesin, and there is one cytoplasmic dyneem. So in that respect, what happens to dyneem can be more important rather than what happens to one of those 45 kinesins.
Right.
Right.
So many kinesins could also in that way play together to cause these kind of disorganizations.
Surely they could. So there are so many different kinds of kinesins. Nobody understands all the kind of things that I told you about this particular kinesin. It may be different for another kinesin.
Right. Okay. Thank you.
Hi, Roop. I have one quick question.
Yes,
so, related to the current slide. So here, what I understand is, so this clustering of dyneem happens with an increase in cholesterol. So,
yes.
Typically, what is the time scale of such process? And
minutes. 10 minutes, approximately. Depends on cell to cell type. Minutes. Yeah.
Okay. And what is the exact mechanism, like the interaction between cholesterol and these motors, which leads to this kind of clustering?
Right. So I can't give you a 100% sure answer, but very likely that cholesterol, there is a particular protein called ORP1L. The name is not important, but that protein has a cholesterol sensing domain. Okay. Cholesterol basically pokes out of the membrane, and this protein has probably a hydrophobic domain which recognizes this and goes and binds to cholesterol. Okay. And dyneem goes and binds to proteins which bind to ORP1L. So that's the best I can give you right now.
Okay.
But what we did show, because you asked that, is of leishmania, right? What we did show that a particular lipid from leishmania, in this paper we have shown that a particular lipid from leishmania actually blows apart these cholesterol rich domains. Okay. Because it has an even higher binding affinity to these cholesterol rich domains, and very likely when it comes in, it brings so much electrostatic charge with it that it blows apart these domains, and that might be one way in which leishmania avoids going to the phagosome.
Okay.
But that's kind of speculative. You can read the paper. Yeah.
Okay, sure. Thanks.
Yeah.
Hello, Professor Ruth.
Yes.
Yeah, I have one basic question, that how this lipid droplet decide that how much time or they have to interact with the membrane, or how much cargo they have to release to the cell.
How do motors know how much time they have to interact with the membrane?
No, you have shown that lipid droplets interact with the membrane. Fusion of lipid membrane takes place. So
I cannot hear you very well. Sorry, maybe something wrong with my audio.
So can you hear me now?
Yes. Yeah. Can you repeat your question?
Yeah. So like lipid droplets fuse with the membrane. So how they decided how much time they have to stay there for fusion, and or how much amount of cargo they have to release? Do they release all the cargo, or it's like sort of like there is some?
No, these interactions are all very dynamic. There are certain, given a composition of the membranes, there is a certain time for which this membrane and that membrane will interact, because essentially, given the membrane composition, there is a binding energy, and once there is a binding energy, you can calculate a characteristic time for that event, right? So it may bind transiently and come off, or it may bind on longer time scales. So once you calculate the binding energy, you can estimate a time scale for these things, right?
Oh, okay.
Yeah.
And do these lipid droplets interact among themselves?
Yes, absolutely they do. They can fuse also. Yes.
Okay.
So that's why in your adipose tissue, in your fat tissue, basically your fat cells are very, very large lipid droplets.
Okay. So
And
like
that's why you can that's why you can sit down comfortably on a chair.
Okay, so like there's some sorts of like communication, or what decide that how much they will fuse, or it is a random process, like 4 or 5 they will fuse, or like, or just I am curious to know.
No, who will choose who?
Like how they decide that, okay, how much they have to fuse, or how much lipid droplets have to fuse, or like it's a random process.
I don't have a complete answer, but one possibility is that the proteins which decorate the membrane, they, whether, here is one lipid droplet, here is another lipid droplet, okay?
Okay.
Now there is some protein attached to this and some protein attached to this. Depending on these proteins may want to interact like this, and then these lipid droplets will come together. There may be lipids on this, which like phosphatidic acid. It is known that if you increase phosphatidic acid, you start making huge lipid droplets. Okay. So all this is known, but if I take names of proteins, that's not very useful.
Okay.
So are there any more questions? So if not, let us thank Professor Roop Mallik for this wonderful talk. And we will, so next colloquium, we will have Professor Gerhard Gomper. So that will be on 11th of May. So thank you all for actively participating, and hopefully we'll see you again next month. Thank you, Ruth.
Thank you, Miten. Thank you, everybody. Bye. Yeah.